So for our last bit on cellular respiration, we are going to discuss meiosis. So when we think here, and we're looking in this particular case, we are looking at sexual reproduction. Asexual reproduction is essentially just mitosis, which we've already done. In sexual reproduction, offspring acquire genes from the parents by inheriting chromosomes, right? My sperm and ova combine in the nucleus of a fertilized egg, and all of that information is combined. Now, a chromosome is considered an organizational unit of hereditary material. So we have all these different genes, and each gene is located at a very specific region or locus on the DNA molecule. For example, I'll just go sex-linked for a second because those are the easiest ones. Your red-green color blindness is found on a certain region of the X chromosome. Okay, hemophilia is found on a certain region of the X chromosome. Male pattern baldness is actually found on a certain region of the X chromosome. The gene that I researched during my graduate career was found at the very end of the P arm of chromosome number 17. Okay, so we know where genes are specifically located. Many genes, in fact, are specifically located. So each of our somatic cells has 46 chromosomes, which come in matching pairs of 23. Okay, the only ones that don't always match are the sex chromosomes. So you could have two X's or XY. Okay, this individual is a male, and I can tell this because this is the Y versus the X chromosome. This is also why when we get into it, the existence of sex-linked genes is you'll notice that the Y chromosome is considerably smaller than the X chromosome, which means that the Y chromosome doesn't carry it doesn't have all the same genes on it, so it doesn't balance out all the time. Okay. And then anything that is a non-sex chromosome is referred to as an autosome. Gametes have a single set of 22 autosomes and a single sex chromosome, and they are considered haploid. You need to know that word, okay? Haploid is really important. Diploid is when you have the 2N, okay? The correct number of chromosomes, essentially, in humans is diploid. Okay. So this is just a cute little, if 2N equals 26 on my little froggy here, N is going to be 13. Dolphins have 44, N is 22. Drosophila melanogaster, our happy little fruit fly, right? N is 4, and our tomatoes, N is 12. Okay, that's all there really is to it. So meiosis reduces the number of chromosomes. I go from a diploid number to a haploid number. This increases genetic variation among offspring, which we'll get into in a great deal in just a moment. Single replication of DNA followed by 2 consecutive cell divisions. Okay, so I undergo G1S and G2 like normal, but then I go basically through 2 divisions, and it produces 4 genetically different haploid daughter cells. Okay, mitosis produces genetically identical diploid daughter cells. So during the first division and in meiosis 1, homologous chromosomes pair. So if you remember the last one, I talked about chromatids and how we were going to have all these different ones. So if I look here at my 2 dark blue chromosomes, these are sister chromatids. Okay, my 2 light blue ones are also sister chromatids. They are genetically identical to one another. My light blue and my dark blue are homologous chromosomes. Translation, homo meaning the same, right? But in this case, it means that they're both, say, for example, chromosome number 6. Okay, when pairing up happens, chromosome 6 all pairs up together. It's not chromosome 1 with chromosome 23. Okay, so that's what we mean by homologous chromosomes. However, homologous chromosomes are not necessarily genetically identical to one another. For example, for me, eye color gene, and I'm not remembering what chromosome it is on, but my mom, I have a blue-eyed gene from her, and my dad, I have a hazel-eyed gene from him. So these 2 would both contain the blue-eyed gene. These 2 both contain the hazel-eyed gene. They're homologous because they both contain the eye color gene, if that helps. The alleles are not necessarily the same. So the allele meaning the version of the gene that I have, blue versus hazel. While paired up, they cross over and exchange genetic information. And crossing over is a huge, huge thing in making it such that we now have more genetic diversity. Okay. They then separate and two daughter cells are produced. So my homologous pairs separate. Notice how I've still got my kinetochore here. Like my sister chromatids did not separate. Okay, they're no longer sister chromatids because they're no longer genetically identical, but you get what I'm saying, hopefully. And if not, we'll ask me about it. Anyway, so that you'll notice that they then separate and those two daughter cells are produced. Okay, so this is what we're doing here. I'm doing my crossing over. Okay, these then, they do line up along a metaphase plate. Okay, but notice how there are basically two on each side instead of just one on each side. Okay, they separate. During interphase 1, my chromosomes still replicate. Okay, this is the same as it was with mitosis. Synthesis occurs and I duplicate all of my genetic material. Okay, I've got my centrioles, I've got my centrosomes going on because, and again, we don't know what they do, but they're there. Okay. During prophase 1, chromatin condenses. Synapsis. Synapsis is another word for crossing over. You need to know that they're synonyms. Okay. Only happens in prophase 1. Please repeat after me. Synapsis only happens in prophase 1. Okay. It's really, really important. I'm going to ask you about it on the test. Here, the chromosomes can be seen as distinct structures. The way, and I didn't do the analogy earlier, and I ought to have when I was talking mitosis. The way I think about it is this. When we're happily living someplace, all our crap is strewn all over the place, but when we go to move, we pack it into boxes because it makes it easier. Okay, that's essentially what DNA is doing here. We want easy access to it. I don't want my glasses in a box that I'm digging through to try to find. I want them in my kitchen cupboard where I can find them. Okay, I don't know if you guys have ever moved, but when you move to college, it's a pain, okay? But you pack up everything and then you unpack it again. DNA is usually unpacked so we have access to the information that we need. But when we go to move, we want to pack everything up. Okay, so each chromosome has two chromatids. So this synapsis forms a tetrad, this T-A-T-R-A, right? Tetra, meaning 4. Okay. Sister chromatids are held together at the centromeres. Non-sister chromatids are held together by chiasmata, which is where the crossing over occurs or the exchange of DNA. Okay, so this happy little DNA swapping is going on. This is just another diagram of it. I'd like to point out that DNA, you can literally have crossing over occur at 20 different places along the side of the chromosome. When we're showing it in animation, it's just a lot easier to see it. But notice here, I really want to draw your attention to the bottom. I now have, these are all going to separate, and I'm going to have 4 genetically different daughter cells. Okay, my centriole pairs move apart. I've got my spindle fiber formation. I have my nuclear envelope disappear, just like in mitosis. Okay, so this is just prophase 1. It's a big, old, globbly, gooky mess. During metaphase 1, my homologous chromosomes pair up along the plate. Again, like I showed before, we're noticing that there are 2 on each side instead of just 1 straddling it. My metaphase plate isn't going in between the kinetochore, it's going in between these homologous pairs. Okay, it's breaking up the tetrad. Again, I've got this fun little sort of Sharpie marker thing going on. In anaphase 1, my homologous chromosomes separate independently from one another. Okay, and this will happen, we're only showing 2 sets of chromosomes here. In humans, we've got all 23 pairs. Okay, so this is anaphase going on. You can see they're sort of pulling apart there. And then telophase 1 and cytokinesis. Each pole now has a diploid set of chromosomes. Okay, each with 2 sister chromatids. Usually we get cytokinesis going on here. I've got my cleavage furrow or my cell plate forming. Okay, plants do reproduce sexually. Most of them, many plants do reproduce sexually. So yeah, this is going on in plants too. Okay, this is telophase 1. You can tell because they're really far apart. Remember, telo means far, okay, like a telescope. And then meiosis 2 is essentially the same as mitosis. Now, I'm not going into interphase again. I don't re-enter interphase. I basically go from telophase 1 / cytokinesis right into prophase 2. Okay. Here, my sister chromatids of each chromosome separate. And then the result is 4 haploid daughter cells. Okay. So this is meiosis 2. No, now both of these cells are going to go through the same process. Again, now I've got my metaphase plate shooting right through my kinetochore there. Okay, and then they separate out, and then I have 4 separate daughter cells. So prophase 2, my spindle apparatus reappears, chromosome move toward the metaphase plate. You'll note that they don't say anything about the nuclear envelope here. The nuclear envelope does not reform between the 2 phases of meiosis. That would bluntly be a waste of energy, and Mother Nature is nothing if not efficient. Okay, so there's prophase 2. Metaphase 2, my chromosomes align. Again, notice how now it's sort of this, it's a picture of what we usually consider, like when I think of a chromosome, I think this little 4 thing here, right? Okay, and it's straddling the metaphase plate. Again, my Sharpie marker. Anaphase 2, the sister chromatids are now separated and move towards opposite ends of the cell. Okay. And then during telophase 2, I've got my nuclei reform. Okay, my nuclear envelope starts showing up, my cytokinesis occurs. Okay, my cleavage furrow in animal cells, my cell plate in plant cells, all of this happens, and now I end up with 4 unique haploid daughter cells. Okay. Key differences between mitosis and meiosis. This is important. Okay, meiosis is it's involved in reproduction. It's a reduction division. Mitotic cells produce clones. They have the same number of chromosomes. Okay, when my skin cells divide to repair the cut that I have, I still have 46 chromosomes. Meiosis reduces it. Okay, I now have haploid cells. I now have only 23 chromosomes in each. Meiosis produces genetic variation. Okay, it produces 4 genetically different daughter cells, whereas mitosis produces 2 identical daughter cells. Meiosis is 2 successive nuclear divisions, whereas mitosis actually only goes through 1 division. Okay, this is what's going on. This picture here, sort of, you guys are going to be drawing for me. I will give you the date specifically if you guys can make it. My people that are more virtual, if you guys can make it that day, that's fabulous. Otherwise, we will Zoom the whole time so that you can draw. And everybody hates me when it starts, but I promise you that it will make more sense in the end. So genetic variation occurs in a couple of different ways. Crossing over, right? So now it's not like your gametes are all like, okay, I've got a gamete that has like all of dad's chromosomes in it and all of mom's chromosomes in it. It's they're crossing over, okay? And by crossing over, I shake things up a bit. Independent assortment, each pair of homologous chromosomes separates independently. So again, it's not like you're going to end up with a chromosome that is, or you're not guaranteed to end up with a gamete that is exclusively all the chromosomes that came from one parent. You know, crossing over only occurs on 2 of the chromosomes, or on 2 of the sister chromatids in each pair. So it's not like only mom, mix, only dad. Okay, that's just this independent assortment allows for this variation. Okay, and then random fertilization, you know, it's may the best man win. Okay, there isn't necessarily a rhyme or reason to which sperm gets there first, which ovum actually gets fertilized. So it's the random joining of 2 gametes. Crossing over, this is an electron microscope showing of crossing over. So as I said, typically when we draw it, we really only do one or two spots, where it really happens on multiple loci. Okay, again, only during prophase 1. So this just, again, is a nice way to show, okay, wow, here we go. I've got, these different parental chromosomes. I've got these recombinant types going on. We have co-color gene versus I-color gene. And now all of a sudden I've got offspring that, might look different. Okay. So mom, I'm going to go with this, by the way, hair color, eye color isn't this simple in humans, but you might say that you've got mom is blonde haired, blue eyed, and dad is dark haired, brown eyes. And you might end up with a dark haired, blue eyed child. Okay. If dad happens to have also the gene for blue eyes, it's just, you get the variation. Okay. This is independent assortment. Okay. This is just showing how you don't necessarily, it's not always going to end up the same. These chromosomes, these homologous chromosomes separate independently from one another. Okay. And again, this is just showing another example of it. And this is also independent assortment. And then random fertilization, right? So genetic variation is really important because it is what leads to evolution. Okay. New combinations are what lead to evolution. We have some traits that are favorable and some traits that are unfavorable. Okay. Now, I'd like to point out, and we'll get more into mutations later when we really get into Mendelian genetics. You can tell I'm really excited about that. Anyway, the thing is, mutations get a bad rap. Mutations aren't inherently bad. Okay. Mutation simply means change. We tend to think of mutations being negative, but mutations can be very positive too. Now, errors and exceptions. Okay, things that can go wrong. We can get alterations in the number of chromosomes or the structure that can cause disorders. So I can have this happen, physical or chemical disturbances. This is why they put a lead vest over the important parts of your body whenever you have a X-ray, so you don't end up with Blinky the three-eyed kid. It is to protect your gametes. When you talk about radiation, things like that, you want to make sure you're being careful. Also, errors during meiosis. Okay. To study human chromosomes, we usually take them out. We can essentially, you can extract it, and then you stain it, and they're displayed as a karyotype. You're going to do an activity with this. A karyotype usually shows 22 pairs of autosomes and 1 pair of sex chromosomes. Obviously, this would be a human karyotype. Okay, so I do the centrifuge. I hypotopic, right? Or hypotonic, not topic, sorry, which causes the cells to burst. And then I fix them, and then I stain. Now this is much nicer than the one you guys are going to be doing, because this is done with fluorescent in situ hybridization fish. Okay, this is a lot easier. The ones you guys are doing are in black and white. You're going to have fun with it. It's awesome. Okay, this is what you guys are going to be looking at. So this is the female banding. Okay, and then you essentially, like, seriously, like, you cut them out and you match them up. And how we match them up is actually looking at the banding pattern. Okay, so for example, I'm going to look here. This chromosome and this chromosome would pair up. Okay, part of the reason I can tell is this very, very thick band in the center. Okay, and not so much the banding on the other ends. Okay, so that's what you're just looking to see, where these light and dark bands line up. It takes some time, admittedly. Okay, it takes a little bit of getting used to in order to do it. And this we know is female because of 2 X's. Okay, again, we would cut it all apart, and this is male because this individual is male because we have XY. So non-disjunction. When I have a pair of homologous chromosomes that do not move apart during meiosis 1, or sister chromatids that do not separate during meiosis 2, this can result in an uneven distribution of chromosome and daughter cells. And this leads to aneuploidy, which is an abnormal number of chromosomes. Trisomy is 3 copies of a chromosome. Monosomy is 1 copy of the chromosome, and most of these are lethal. Okay. In fact, you've got triploidy of 17, 18, and 21. 21 is Down syndrome. So 3 copies of 21 is Down syndrome. 17 and, sorry, I'm going to look that up before we get talking again. Monosomy is only 1 copy of a chromosome. This very rare, the only one that I'm honestly aware of off the top of my head is Turner syndrome, which is when you have a female that only has 1 X instead of 2 X's. If you guys have ever watched NCIS LA, the woman who plays Hetty, who is also the voice of Edna Mode in The Incredibles, actually has Turner syndrome. Okay, so this is where we've got non-disjunction going on here. You'll notice I've got 3 copies here and only 1 copy here. Okay, this is if it happens during meiosis 1. And then fertilization after non-disjunction, I then end up with extra chromosomes or lacking a chromosome. Okay, trisomy 21, which is Down syndrome, is sort of our poster child for trisomies. They're short stature. We have that typical heart-shaped face. They can have very, very severe heart defects. Most common of the serious heart defects, it's about 1 in every 700 births. Mothers over the age of 35 have a much higher chance of having Down's baby. I will talk about that more later when we get into some other stuff as to why. There's actually a pretty strong hypothesis as to why that happens. And this is just showing that Down syndrome goes up with maternal age. This is a Down syndrome karyotype. Right there, 3 copies of chromosome number 21. Okay, non-disjunction with sex chromosomes. These are much more prevalent and there are some issues associated with it, but not too many. So in Klinefelter is XXY. Klinefelters, individuals that have Klinefelters, they are male. They present as male, but as they go through puberty, you get some more female secondary sex characteristics, like they have a little bit more breast development. And it can impact cognitive ability. There are varying degrees of mental retardation that can come with it. Most people with Klinefelters are perfectly well functioning, but you can have more severe cases of it. XYY, some people refer to this as Superman syndrome. It's 1X, 2Y chromosomes. There was a theory at one point in time that these men were more likely to be murderers, rapists, etc., because they thought perhaps the extra testosterone being produced by the Y chromosome. In fact, they did lots of studies. They took the genetic information from many prison inmates, and there was no correlation at all. Metafemale is XXX. And so they develop as female. I believe that they are sterile. And I know for an absolute fact that females that are X not, so there's only 1 X chromosome, are absolutely sterile. They also tend to be short in stature and have some other issues, some other defects and whatnot that go along with them. We have others. Breakage of a chromosome can lead to 4 different types of changes. I've got a deletion. Okay, part of it is lost during cell division. A duplication, I may actually get an extra little bit in there. Inversion, where I flip it, or translocation, where I actually move to a different location. So this is a deletion. This is a large region deletion. I'd like to point out most of them aren't this big. Okay, and this actually causes Creux-de-Chat syndrome. It's, for those of you who took French, this means cry of the cat, because when they cry, they sound like a cat, literally like the babies sound like a cat meowing. It has a lot of major implications with it. And it's a partial deletion. So 5p, P is the short arm of the chromosome, okay, as opposed to the long arm of the chromosome. Inversion, you can see here, this thick band is now down here and these 2 thinner bands are now at the top. Translocation, all of a sudden I've taken this and put it in here. Okay. This is what causes CML, or this is one thing that can cause CML, which is chronic myelogenic leukemia, is it is actually a translocation. And that is it.